Good morning, everyone. I'm Dr. Preeti Sharma, Professor, Department of Biochemistry. Today I'll be talking on xenobiotics metabolism. A xenobiotic is a compound that is foreign to the body. Xenobiotics can produce variety of biological effects, including pharmacological responses, toxicity, immunological responses, cancers. Xenobiotic can be exogenous or the endogenous. So in the exogenous, most of the time they are exogenous. In our day-to-day life, we meet with a lot of exogenous substances in the form of number of dietary foodstuff or in the form of certain medicines, drugs used for therapeutic cause or are inhaled through environment. For example, drugs, food additives, different pollutants coming out from the industries and insecticide chemical carcinogens. As far as endogenous xenobiotics are concerned, though they are not actually the foreign substances, but they possess similar effect like those of xenobiotics, which are exogenous in nature. These are synthesized in the body or are produced as metabolite of various processes in the body. For example, bilirubin, bile acids, steroid, eicosanoids, and certain fatty acids. Metabolism of xenobiotics. For better understanding of what can be the fate of xenobiotics once you take in your body intentionally or accidentally, then what can be how the body treats these xenobiotics. So for a better understanding of its metabolism, the process of metabolism has been divided into two segments. The phase 1, that is the major reaction involved in is the hydroxylation. In addition to the hydroxylation, a wide range of reactions are also taking place, including deamination, dehalogenation, desulfuration, epoxidation, peroxygenation, reduction. Phase 1 reaction enzyme, mainly catalyzed by the members of class of enzymes referred to as monooxygenase, mixed function oxidases, cytochrome P450. Other enzymes of significance are aldehyde and alcohol dehydrogenase, deaminases, esterases, amidases, epoxide hydrolases. All the biochemical reactions involved in the conversion of RM toxic and water insoluble molecule to the non-toxic, water soluble and excretable forms are called detoxification or the biotransformation reaction. Overall purpose of these two phases of metabolisms of xenobiotics is to increase the water solubility and thus excretion from the body. In certain situations, these reactions may instead increase the toxicity of foreign compound. Then these are called intoxication reaction. That means our body is not aware of whether the biotransformation which it is going to do will lead to toxic or non-toxic metabolite. Biotransformation reaction. The purpose of the reaction is only to convert the lipophilic to hydrophilic compounds, facilitate the excretion. Consequences: change in solubility characteristics, detoxification, metabolic activation. The main organ involved in all these xenobiotics reaction, especially in the phase 1 reaction, is your liver. Liver contains enzymes concerned with phase 1 biotransformation. Hepatocytes contain wide variety of enzymes to process xenobiotics. Enzymes are present in cytosol, endoplasmic reticulum, and to the lesser extent in other organelles. Each enzyme represents a large family of gene products. Each gene product may be induced by different xenobiotics. Overview of biotransformation reaction. The phase 1 reactions can limit the toxicity of a drug. The phase 1 reaction can also convert the xenobiotics from inactive to biologically active compounds. In these instances, the original xenobiotics are referred to as prodrug or procarcinogen. Phase 2 or conjugation reactions can convert the active product of phase 1 reactions to less active or inactive species, which are subsequently excreted out of the body through urine or blood. In a very few reactions, the conjugation may actually increase the biological activity of the xenobiotic. So the overall purpose of xenobiotic metabolism, it must be clear to you all, to convert the molecule into something which is inactive to some active product. Say for instance, suppose you have working in the laboratory where the benzene fumes are coming out. Benzene itself is a very hydrophobic substance. So, but once it goes to the body won't leave it as such, because it is being hydrophobic, have longer span of stay in the body, because it is not miscible with the transporting system of the body. So it will stay in the body and it may cause toxicity. So in order to cope up with this, the body has this advanced system of phase 1 metabolism, especially in the liver. The enzymes work on this, and the phase 1 and cytochrome P450 enzymes convert this molecule into more hydrophilic kind of entity by addition of some functional group. So phase 1 reactions can limit the toxicity of drug, or they may also increase the toxicity of drug. So what can be the consequences in nutshell? The biotransformation may lead sometime the potentially toxic xenobiotics into the inactive metabolite, or sometime relatively harmless substances can be converted into the reactive substances or reactive metabolite. So in this previous kind of conversion, it is known as detoxification, or it may be metabolic activation. This is the overview of detoxification reaction. So this is, suppose a drug as a xenobiotic, it is entering into the body, it meets with the phase 1 enzymes. It may have many fates depending upon the type of entity present in the chemical. So it may undergo oxidation, it may undergo reduction, it may undergo hydrolysis, but more than 90% kind of reaction, there can be generally hydroxylation kind of reaction happen. So once it is added with any entity, if it is added with the hydroxyl group in itself is very polar, and it makes something which is hydrophobic to convert it into a more hydrophilic entity. So this is the purpose of phase 1 biotransformation. Further, if the generation of hydrophilicity is not enough to remove it out of the body, then the molecule or then the metabolite will undergo phase 2 biotransformation. Under phase 2 biotransformation, there will be conjugation reaction, and the conjugating substance will in itself will be very, very inactive, and as a consequence of phase 2 reaction, now it is in a form which can easily be excreted out of the body. Or sometime the phase 1, the drug is enough hydrophilic, it has some hooks in the form of some hydroxy group or some functional group, drug may escape the xenobiotic may escape the phase 1 biotransformation. It can directly undergo the phase 2 metabolism. So they can be the varied fate and pathway of phase 1 and phase 2 biotransformation. Sometimes drug or xenobiotic substance is enough hydrophilic, so after a certain or small stay in the body, it will automatically removed out of the body. It can skip phase 1 and phase 2. There is no need of conversion because its bioavailability in the blood is very, very less. Then comparing the phase 1 and phase 2 reactions, these are the types of reaction. So phase 1 undergo hydrolysis, oxidation, reduction. Phase 2 are generally conjugation reaction. Next is increase in hydrophilicity. There is a small increase in hydrophilicity as compared to the phase 2. The general mechanism, it exposes the functional group and the polar, in this phase 2 type, the polar compounds added to the functional group. What can be the consequences? It may result in metabolic activation, or in this segment it facilitates the excretion of metabolites. Now, what can be the factors affecting the biotransformation of the drug? They can be prior administration of a drug or co-administration of other drugs. So that is a drug-drug interaction, which you will subsequently read in pharmacology section. Then diet can have an effect onto the biotransformation, then hormonal status, then genetics, diseases, age, developmental status, functional status of liver and kidney. All these factors may impact onto the xenobiotic metabolism of xenobiotics. Let us have comprehensive look onto the phase 1 reaction. The phase 1 reactions include the oxidation, reduction and hydrolysis reactions. They are also called the hydroxylation reaction. Since they introduce or expose a functional group, they serve as an active center for sequential conjugation in phase 2 reaction. Let us give one example. Suppose this is methyl group is oxidized to acid through formation of alcohol and aldehyde. So this is the general fate of any methyl containing group. So methyl can be converted to is introduced within hydroxyl group. Hydroxyl can further change to aldehyde, and aldehyde can further change to acidic group. This is the methanol. Methanol, once it is being consumed, it's being said, it produces toxicity. So the toxicity can be due to the conversion of methanol to formaldehyde, and formaldehyde converts to formic acid in vivo under the influence of cytochrome P450 enzyme, and this toxicity produced by methanol is not because of methanol itself. It is because of formation or conversion of methanol to formic acid, which is very, very toxic to the body. You can see this is methanol and it is undergoing conversion under the influence of enzyme alcohol dehydrogenase. Though it is not cytochrome P450 enzyme, but the same kind of a conversion it manifests. So, and formaldehyde further converts to formic acid. And this formic acid further converts to, it may cause metabolic acidosis and tissue injury. Enzymatic reaction will be worstly affected under the acidic pH. Further, it may convert into smaller molecule carbon dioxide and water. This is another example of oxidation of benzene. So benzene may undergo hydroxylation. Similarly, the benzaldehyde, it converts to benzoic acid, which is more hydrophilic. This group is more hydrophilic as compared to this aldehyde. So this is better, this is the best example going to show you that it is following the principle of nature, like converting something which is hydrophobic into the more hydrophilic entity so that it can easily be excreted out of the body. So it's for longer stay, it may produce toxicity. We can take example of acetyl salicylic acid or aspirin. It converts after the hydrolysis into acetic acid and salicylic acid. Similarly, atropine and tropic acid and tropine. So this aspirin is a kind of analgesic, and analgesic activity is not because of acetyl salicylic acid, it is because of salicylic acid, which is produced once this drug is being metabolized under the influence of hydrolytic enzymes of cytochrome P450. Similar fate is that of procan. Procan converts to para-aminobenzoic acid and diethylaminoethanol. Similarly, diacetalates converts is converted into sugar and egg glycol. Now, what are actually the enzymes? Cytochrome P450 heard many a times, but actually what are the contents present and how it works? Mainly catalyzed by a member of class of enzyme referred to as monooxygenases or mixed function oxidases or cytochrome P450. Other enzymes of significance are aldehyde and alcohol dehydrogenases. Then we have deaminases, esterases, amidases, epoxide hydrolases. Cytochrome P450 enzyme system, the reaction catalyzed by the monooxygenases is as follows. This is suppose this is some xenobiotic, and xenobiotics require the molecular presence of oxygen gas, NADPH as a reducing equivalent and a source of hydrogens. So in this course of reaction, what happens? The xenobiotic converts into a more hydrophilic entity, that is the ROH. Now initially there was a hydrogen, so hydrogen is being replaced by a hydroxyl group. So obviously because of inclusion of this hydroxyl group, there is an addition of polarity and the molecular becomes more polar. So like alcoholic kind of molecule which is easily or nicely miscible with the aqueous system of the blood, and it can now easily be excreted out, plus water and NADPH. So one thing more, sometimes this enzyme is also known as mixed function oxidases. So mixed function of oxygen. Here oxygen has dual function. At the one atom of the oxygen is inserted into the xenobiotic in the form of hydroxy group. Another oxygen atom is going to be reduced and forming the water molecule. So there are dual fate of oxygen. That's why these enzymes are also known as dioxygenases and or the mixed function oxidases because of the dual or mixed function of oxygen. RHs above can represent a wide variety of xenobiotics, including the drugs, carcinogen, pesticides, petroleum products, etc. In addition, endogenous compounds such as certain steroids, icosanoids, fatty acids and retinoids are also substrates. The substrates are generally lipophilic and are rendered more hydrophilic by hydroxylation. Now, properties of human cytochrome P450 involved in phase 1 metabolic reactions of innumerable xenobiotics. It is involved in the metabolism of many endogenous compounds. All are heme protein. All of these contain a heme ring in their scaffold, exhibit the broad substrate specificity, thus act on many compound, extremely versatile catalyst, perhaps catalyze about 60 type of reaction. Basically, they catalyze the reactions involving introduction of one atom of oxygen into the substrate and one into the water. Liver contains the highest amount but found in most, if not in all the tissues, including the small intestine, brain and lung, located in a smooth endoplasmic reticulum or in the mitochondria. In some cases, the products are mutagenic or carcinogenic. Many have a molecular mass of 55 kilodalton. Many are inducible, resulting in one cause of drug interaction. Many are inhibited by various drugs or their metabolic products, providing another cause of drug interaction. Some exhibit genetic polymorphism which can result in atypical drug metabolism. So that's all about the introduction about the phase 1 reactions, the concerned enzyme cytochrome P450. So in the next lecture, I'll be continuing about the phase 2 conjugative reactions. Thank you so much.